Okay, let's unpack this. Imagine those subtle rhythmic movements happening constantly inside your body.
Hmm.
You know, the way food moves through your digestive system, how your blood vessels adjust pressure, even your pupils dilating in a dim room.
Right, things we barely notice.
Exactly. These aren't random acts. They're orchestrated by this incredible, often overlooked tissue called smooth muscle.
That's right.
Today, we're not just scratching the surface. We're aiming for a really deep dive into the world of smooth muscle.
Yeah, let's get into it.
We'll unravel its unique structure, its surprising ways of contracting, and why truly mastering this information is, well, absolutely
It
crucial.
really is, from basic body functions right up to developing new treatments.
Our goal here is to go as deep as we possibly can, cover every concept so you can really understand this stuff like the top professionals in the field.
And what's fascinating really is just how ubiquitous smooth muscle is. It's a major part of nearly every hollow organ you can think of.
Like where specifically?
Well, the whole alimentary canal, your airways, the entire vascular system, the urogenital tract,
Yeah.
it's everywhere.
Okay.
And its fundamental job is to precisely alter the dimensions of these organs. So that could be propelling contents along, like in
Right,
peristalsis.
the waves in the gut.
Exactly.
Yeah.
Or increasing resistance to flow, like when blood vessels constrict, vasoconstriction.
Which is critical for blood pressure.
Absolutely critical. Now, you're probably familiar with skeletal muscles, the ones you consciously move, and the heart's cardiac muscle.
Sure.
But smooth muscle, it operates with its own distinct rules, often far more nuanced and complex than you might expect.
So our mission today is to really grasp those rules.
That's the plan, a proper deep dive.
That's a great setup because, yeah, when we think muscles, it's usually conscious control, like lifting something or the heart's automatic rhythm. Smooth muscle feels different.
It does.
So fundamentally, what makes its contraction mechanism so different from, say, skeletal or cardiac muscle?
That's the perfect place to start. The core is still myosin interacting with actin, the basic building blocks.
Okay, the motor proteins.
Right. But the critical difference, the main concept, is that smooth muscle contraction is thick filament regulated.
Thick filament regulated. What does that mean exactly?
It means the myosin itself, the thick filament, needs to be activated before it can even think about interacting with actin.
Activated. How?
Through phosphorylation. It's a chemical switch. Intracellular calcium comes in.
Calcium's always key, isn't it?
Always. Here, it binds to a protein called calmodulin. This calcium-calmodulin complex then activates an enzyme myosin light chain kinase, or MLCK.
MLCK. Okay.
And that enzyme, MLCK, adds a phosphate group to the myosin light chain. It phosphorylates it.
Ah, so phosphorylation is the essential on switch for the myosin.
Absolutely essential. Without that phosphorylation, the myosin head simply cannot effectively bind to actin and cycle to produce force.
That's completely different from what I remember about skeletal muscle.
Totally different. Think of it as a gatekeeper right on the myosin. In contrast, striated muscle, skeletal, and cardiac is thin filament regulated.
Thin filament.
Yeah. There, calcium binds to a protein called troponin, which sits on the actin filament, the thin filament.
Right, I remember troponin.
And that binding just causes the troponin-tropomyosin complex to shift, to move out of the way, exposing the binding sites on actin that myosin wants to grab onto.
So in striated muscle, myosin is always ready, just waiting for the path to be cleared.
Exactly. Calcium acts like a traffic controller, opening the road. Smooth muscle, interestingly, doesn't even have troponin.
No troponin at all. Wow.
Nope. This fundamental difference in regulation, thick filament versus thin filament, allows smooth muscle to respond to a much wider range of signals, not just electrical, but hormonal too.
Okay, that explains its versatility.
And crucially, it lets it sustain contractions for really long periods, hours, even days. And it does this using remarkably little energy.
Low energy cost. That sounds important.
Vital. Absolutely vital for things like maintaining the tone in your blood vessels, keeping blood pressure stable. That constant low-level contraction needs to be efficient.
That deep dive into the mechanism is already super helpful. So, okay, smooth muscle isn't just one thing, it's diverse. How do we start classifying it? What are the main types?
You're right, it's far from uniform. You can think about it on a spectrum, but broadly, we subdivide it into single unit and multi-unit groups.
Single unit and multi-unit, okay.
In single unit smooth muscle, the cells are electrically coupled. They act together like one large unit,
How are they coupled?
typically through gap junctions. These are little protein channels connecting the cells, creating low resistance pathways.
So electricity can flow easily between them.
Precisely. An electrical stimulation in one cell spreads rapidly, like a wave, to all its neighbors. This gives you coordinated synchronous contraction.
Like peristalsis.
Exactly like peristalsis in the intestine. That wave-like motion pushing things along. It's a classic example.
Can these waves start on their own?
Sometimes, yes. Single-unit tissues often have specialized pacemaker cells. These cells can spontaneously depolarize, generating their own electrical rhythm, a bit like the heart's pacemaker, but different mechanisms.
Okay, so single unit is about coordination, mass action, sometimes self-starting. What about multi-unit? Sounds like the opposite.
It is, pretty much.
Mm-hmm.
In multi-unit smooth muscle, the cells are not electrically coupled. Stimulating one cell doesn't necessarily activate its neighbors.
So they act independently?
Largely, yes.
Yeah.
Each cell often needs its own individual neural input. This allows for much finer, more localized control.
Where would you find that kind of precision?
Great examples are the VASTF,
Yeah.
involved in sperm transport, where you need precise control, and the iris of your eye. Adjusting pupil size requires very fine, independent control of muscle fibers.
Makes sense. You wouldn't want your whole iris contracting at once.
Definitely not. Now, there's another way to look at this diversity based on their activity pattern.
Okay.
We talk about phasic versus tonic smooth muscle.
Phasic and tonic.
Phasic smooth muscle shows rhythmic or intermittent activity. It contracts and relaxes in bursts. This generally lines up with the single unit type.
So the gut wall again, maybe the bladder?
Exactly. Walls of the GI tract, the urogenital tracts, they contract in response to these propagating action potentials to move contents along. Phasic activity.
And tonic sounds like tone.
You got it. Tonic smooth muscle is continuously active. It maintains a constant level of partial contraction or tone.
Like our blood vessels.
Perfect example. Vascular smooth muscle, the smooth muscle in our respiratory airways, various sphincters, they all maintain tone.
And how does that continuous activity work? Is it action potentials?
Not usually associated with propagating action potentials. Instead, their level of tone, that continuous partial activation, is often proportional to the membrane potential itself. Small changes in voltage mean small changes in tone.
More graded.
Exactly. And this tonic type, which often aligns with the multi-unit structure, is incredibly good at maintaining force at a very low energy cost. Again, that efficiency comes up.
How does it manage that low energy cost?
It's due to some fascinating changes in how the myosin cross bridges cycle. And this leads us perfectly into a really cool property we need to discuss later, the latch state.
The latch state. Okay, put a pin in that. It sounds important. So knowing the types, let's zoom right in on the cells themselves. How are they actually built and connected?
Right, the architecture is key. Smooth muscle cells usually form layers around hollow organs.
Like wrapping around a pipe?
Sort of, yeah.
Yeah.
In blood vessels and airways, they're mostly arranged circumferentially, wrapping around. Contracting them reduces the diameter, increases resistance to flow.
Makes sense.
The GI tract is a bit more complex. It usually has both a circumferential layer and a longitudinal layer running along the length.
Why two layers?
They work together. The circumferential layer constricts to mix food. The longitudinal layer shortens segments to propel it forward. It's all coordinated by those intricate nerve plexuses within the gut wall.
Clever design.
And then you have saccular structures, like the bladder. They often have more varied, almost mesh-like arrangements of muscle fibers. This allows them to stretch and hold a lot of volume without immediately generating strong propulsive force.
It allows for storage.
Exactly. Now, a really crucial structural point. Unlike skeletal muscle cells connecting to tendons,
Right, transferring force to bones.
smooth muscle cells connect directly to each other. This cell-to-cell contact is absolutely paramount.
Why is it so critical?
Think about it. If one cell contracts but isn't mechanically linked to its neighbor, it would just bulge out, maybe stretch the neighbor, without actually squeezing the organ or reducing its volume effectively.
The force wouldn't be transmitted usefully.
Precisely. So these cells need both functional electrical linkage and mechanical linkage.
Okay, and we mentioned gap junctions for the electrical part.
Right. Gap junctions provide that functional and electrical connection.
Yeah.
They're the low-resistance pathways for signals to spread, especially vital in single-unit muscle.
And the mechanical link, what holds them together physically?
That's the job of adherence junctions. You might also hear them called dense plaques or attachment plaques. These are the mechanical anchors.
How do they work?
The thin filaments, the actin filaments inside the cell, actually extend right into these adherence junctions at the cell membrane.
So actin is anchored there.
Yes. And since these junctions connect adjacent cells, the force generated by actin and myosin pulling on those internal anchors gets transmitted directly to the neighboring cell. It provides tissue integrity and allows the whole muscle sheet to contract effectively.
Okay, so external connections are covered. Now, inside the cell, we know they don't have T-tubules like striated muscle. How do they manage internal signaling, especially getting calcium where it needs to go?
That's a huge difference, yeah. No T-tubules. Instead, the cell membrane, the sarcolemma, has these rows of tiny flask-shaped inpocketings called caveolae.
Caveolae, little caves?
Pretty much.
Yeah.
They dramatically increase the cell's surface area relative to its volume. But even more importantly, these caveolae are often positioned very close to the internal calcium store, the sarcoplasmic reticulum, or SR.
How close?
Really close, sometimes forming a gap of only about 15 nanometers. It's a very intimate association.
And why is that proximity important?
Because these caveolae are hotspots for calcium regulation. They're packed with calcium-handling proteins, voltage-gated calcium channels, and that important sodium-calcium exchanger, the 3Na plus NAX1CA plus antiporter.
So they're like little control centers for calcium entry and exit.
Exactly. They seem to be specialized regions for rapidly and precisely regulating the calcium concentration right near the SR and the contractile machinery. We even see localized callus cap plus sparks originating near them, brief bursts of calcium release that act as signals.
Calcium sparks, cool. And the SR itself, the main internal store, how does it release calcium in smooth muscle?
The sarcoplasmic reticulum, SR, is the main intracellular calcium reservoir, just like another muscle. It releases calcium into the cytoplasm, a myoplasm, when stimulated.
Stimulated by what?
Usually by neurotransmitters, hormones, or drugs binding to receptors on the cell surface. This triggers signaling pathways inside the cell.
And what channels let the calcium out of the SR?
There are two main players inside the SR membrane. One is the ryanodane receptor, RYR. Often a little bit of calcium coming into the cell through channels in the sarcolemma triggers these RYRs to open, releasing more calcium from the SR. That's called calcium-induced calcium release.
Okay, calcium triggering more calcium?
Yep. But perhaps even more crucial for many smooth muscle responses is the inositol 1415 tris phosphate NSP3 gated calcium channel.
NSP3, that sounds like a second messenger.
It is. Many hormones and neurotransmitters work by increasing NSP3 levels inside the cell. NSP3 then binds to its receptor on the SR, opening the channel and releasing calcium.
So multiple ways to get calcium out of the store.
Definitely. And then to relax, the cell has to get rid of that calcium. It's pumped back into the SR by the circopump.
Okay.
And also pumped out of the cell entirely by pumps on the cell membrane, like the Canna plus ATPase and that sodium-calcium exchanger we mentioned. It's a constant cycle of release and reuptake or extrusion.
A very dynamic calcium handling system. Okay, what about the actual contractile machinery? No neat striations, so how are the actin and myosin arranged?
You're right, no striations, because the thick myosin filaments and thin actin filaments aren't in that perfect repeating sarcomere alignment you see in skeletal muscle. But there is order. It's not just a random jumble.
What kind of order?
You typically find small groups of thick myosin filaments, maybe 3 to 5, interdigitating with a whole bunch of thin actin filaments surrounding them. These bundles are then anchored.
Anchored to what?
They're connected to structures called dense bodies within the cytoplasm and also to those membrane-dense areas, which are basically the adherence junctions we talked about at the cell surface.
So dense bodies are like the Z lines in skeletal muscle, anchoring points.
Functionally, yes. They serve as the anchoring points for the thin actin filaments. And what's really clever is that these dense bodies are themselves interconnected by the cell's internal scaffolding, the cytoskeleton, specifically intermediate filaments like desmin or vamentin.
So it forms a network throughout the cell.
Exactly. A network linking the contractile units to each other and to the cell membrane. This allows the force generated by actin and myosin pulling on these anchors to be transmitted efficiently across the entire cell, causing it to shorten, often in a sort of corkscrew fashion.
A sophisticated internal network, not just simple lines. Okay, this brings us squarely to control. How are these contractions initiated, modulated, stopped? It sounds way more complex than a simple on-off.
Oh, it's an incredibly intricate symphony of control. Smooth muscle activity is regulated by a huge array of factors.
Like what?
Hormones, signals from the autonomic nerves, local chemical factors, even mechanical stretch itself, plus intrinsic pacemaker activity in some tissues, and of course, various drugs.
A lot of inputs.
A ton. But the common pathway, generally, is that these agents induce contraction by increasing intracellular calcium concentration.
Calcium again.
Always calcium. But here's a really key twist, one of those defining features. Unlike striated muscle, action potentials in smooth muscle are highly variable. Sometimes they happen, sometimes they don't, and crucially, they're not always required for contraction.
Wait, contraction without an action potential? How?
Yes, some agents can trigger calcium release and muscle contraction without causing any significant change in the electrical potential across the cell membrane.
That seems really counterintuitive compared to nerve muscle junctions we usually learn about. What's that called?
It's called pharmacomechanical coupling. Pharmaco relating to drugs or chemicals, and mechanical relating to force or contraction.
Pharmacomechanical coupling.
It means the muscle contracts in response to a chemical agent, like a hormone or neurotransmitter, that doesn't rely on changing the membrane potential.
So how does the signal get transduced?
Typically, the agent binds to a receptor, often a G protein-coupled receptor, on the cell surface. This activates a pathway that increases the intracellular level of that second messenger we mentioned, NSP3.
Inositol phosphate?
Right. And NSP3 then diffuses to the SR and directly opens those NSP3-gated calcium channels, releasing stored calcium.
So it bypasses the need for electrical depolarization to release calcium.
Exactly. It's a direct chemical signal triggering calcium release. This mechanism is incredibly powerful. It allows for very finely graded responses, a little bit of hormone, a little bit of calcium release, and it means a huge variety of substances can precisely control smooth muscle tone without needing big electrical events. It adds a whole layer of regulatory finesse.
Incredible flexibility. So, given that, how do the nervous system and hormones specifically exert their influence? Who are the main players controlling smooth muscle?
Smooth muscle is primarily innervated by the autonomic nervous system, ANS, the sympathetic and parasympathetic branches that operate subconsciously.
Makes sense. We don't consciously control our gut or blood vessels.
Right. For instance, most arteries get mainly sympathetic nerves. Other tissues, like the bladder or parts of the gut, get input from both sympathetic and parasympathetic nerves, often with opposing effects.
And the gut has its own system too, right?
Yes. The enteric nervous system, sometimes called the second brain. It's a complex network within the gut wall that can coordinate much of digestion fairly autonomously, although it's also modulated by the main ANS.
And some tissues don't have nerves.
Some have very sparse or even no direct innervation, like the uterus for much of pregnancy. It relies heavily on hormonal signals instead.
How do the nerve endings connect? Is it like the neuromuscular junction in skeletal muscle?
Structurally, it's simpler and more diffuse. Instead of a single focus synapse or end plate, the autonomic nerve axons have these swellings along their lengths called varicosities.
Varicosities, like little beads on a string.
Kind of, yeah. And these varicosities are packed with vesicles containing neurotransmitters. They lie near the smooth muscle cells, but the gap, the synaptic cleft, is often much wider than in skeletal muscle.
Wider gap. Wow.
It can be 80, 100, even 120 nanometers or more. This allows the released neurotransmitter to diffuse over a larger area and potentially affect multiple smooth muscle cells simultaneously. It creates a more widespread, less point-to-point influence, which fits with controlling a whole sheet or layer of muscle.
A broader signal. Okay, and what are some key neurotransmitters and hormones doing the signaling?
There are many. Let's take norepinephrine, or adrenaline, and epinephrine, adrenaline, from sympathetic nerves or the adrenal gland. They predominantly cause contraction in many smooth muscles, especially blood vessels, by activating alpha-1 adrenergic receptors.
And how do alpha-1 receptors work?
They primarily work through that G protein and SP3 pathway, increasing intracellular calcium.
Okay.
But norepinephrine and epinephrine can also cause relaxation in some tissues, like the smooth muscle and airways or certain blood vessels, by activating beta-2 adrenergic receptors, which work by increasing a different second messenger, cyclic AMP, CMP.
So the same hormone can have opposite effects depending on the receptor.
Precisely. It depends on which receptor types are present on that specific smooth muscle cell.
What about the parasympathetic side? Acetylcholine.
Acetylcholine often causes direct contraction of smooth muscle cells, for example, in the gut or bladder wall, acting on muscarinic receptors. But here's another neat twist.
Another one. Okay.
In many blood vessels, HE actually causes relaxation, but it does it indirectly.
How? Indirectly.
HE stimulates the endothelial cells, the cells lining the blood vessel, to produce and release nitric oxide, NO.
Nitric oxide. The gas.
Yep. NO is a potent signaling molecule. It diffuses from the endothelium into the adjacent smooth muscle cells and causes relaxation, leading to vasodilation. So HE itself doesn't relax the muscle directly there.
That's fascinating. Indirect control via another cell type.
It highlights the integration. And then you have powerful hormones like angiotensin II, vasopressin, endothelin. These are generally strong vasoconstrictors, mostly working via NT3 to raise calcium.
And things that cause relaxation.
Locally produced factors like adenosine, often released from metabolically active tissues, can cause vasodilation, usually by increasing gamma-P, helping match blood flow to metabolic need. It's a complex interplay. Now, let's get into something really crucial for understanding smooth muscle control and dysfunction: calcium sensitization.
Calcium sensitization. Sounds like making the muscle more sensitive to calcium.
Exactly that. Remember, calcium is always required for contraction, but the sensitivity of the contractile machinery to that calcium can be changed. Meaning several hormones and signaling molecules can increase the force of contraction produced at a given submaximal calcium level. If you plotted force versus calcium concentration, sensitization would look like a leftward shift of that curve.
So you get more bang for your calcium buck, essentially.
That's a great way to put it. More force for the same amount of calcium increase.
How does the cell achieve that? What's the mechanism?
A major player here is the Rho-RO kinase, ROK signaling cascade. This is a pathway activated by many of those constricting hormones and neurotransmitters we mentioned, like angiotensin II or norepinephrine acting on alpha-1 receptors.
Rho-kinase or ROK. Okay, what does it do?
When activated, Rho-OA activates ROK, and ROK acts like a double agent, simultaneously doing two key things to boost contraction.
Two things.
First, it inhibits myosin phosphatase, MP. Myosin phosphatase is the enzyme that takes the phosphate off the myosin light chain, causing relaxation. By inhibiting MP, ROK keeps myosin phosphorylated for longer.
So it keeps the on switch jammed on?
Sort of, yeah. It slows down the off switch. And second, ROK can also directly stimulate myosin light chain kinase, MLCK, the enzyme that puts the phosphate on in the first place.
So it promotes the on switch and inhibits the off switch?
Exactly. The net effect is a significant increase in the level of myosin light chain phosphorylation at any given calcium level. This boosts both the number of active cross bridges and potentially how long they stay attached, leading to greater force. That's calcium sensitization.
Wow. That sounds like a really powerful amplification system.
It is, and this is where it gets incredibly relevant clinically.
Because if this pathway is too active...
Precisely. Hyperactivity of this Rho-ARC cascade is implicated in a lot of diseases. Think chronic hypertension, where vessels are overly constricted.
Makes sense.
Think vasospasm after stroke or heart attack, dangerous constriction of cerebral or coronary arteries. Bronchial asthma, where airways constrict. Even things like erectile dysfunction and preterm labor involve inappropriate smooth muscle contraction, potentially linked to this pathway.
So targeting ROK could be beneficial.
Hugely beneficial. There's a lot of research into ROK inhibitors. Studies show they can lower blood pressure, effectively reverse vasospasm in animal models, and even seem to reduce the harmful smooth muscle cell proliferation that contributes to artery narrowing after procedures like angioplasty. It's a really hot area for drug development.
That connection between the fundamental mechanism and potential treatments is fantastic. Okay, so smooth muscle is intricately controlled externally and internally via calcium, but it also has these unique intrinsic properties, right? You mentioned the latch state earlier.
Yes. Let's circle back to that. The latch property is a true hallmark, especially of tonic smooth muscle, the kind that maintains tone. It's all about incredible energy efficiency.
How efficient are we talking?
We're talking about maintaining sustained force, holding that tension, with remarkably low energy expenditure.
Right.
Picture this. During a sustained tonic contraction, the initial calcium spike and myosin phosphorylation peak happens, generating force.
Right.
But then, both the calcium level and the percentage of phosphorylated myosin heads drop significantly. Maybe only 20–30% remain phosphorylated.
Yeah.
Yet the muscle maintains most of the force.
It holds the force even though the activation signal has decreased.
Yes. And because fewer cross bridges are cycling, or they're cycling much slower, ATP consumption plummets. We're talking potentially 300-fold less ATP used compared to skeletal muscle generating the same sustained force.
300 times less ATP. That's astounding. How on earth does it do that?
The exact mechanism is still debated, but the leading idea involves the cross-bridge cycling kinetics. In the latch state, it's thought that myosin heads, once attached to actin, detach much more slowly.
Slower detachment.
Right. One hypothesis is that the myosin light chain can be dephosphorylated while the myosin head is still attached to actin. This latched, dephosphorylated state detaches very slowly, effectively acting like a brake, holding tension with very little ATP turnover. It slows the whole cycle down.
So it enters this super efficient, slow-cycling tension-holding mode.
That's the idea. And it's absolutely vital for functions requiring sustained low-level contraction, like maintaining vascular tone and blood pressure all day long without exhausting itself. This efficiency is why smooth muscle fatigue is rare unless oxygen supply is severely compromised.
Incredible. Okay, what about the other intrinsic property you mentioned, the myogenic response?
Ah, yes, the myogenic response. This is another fascinating intrinsic property, particularly crucial in vascular smooth muscle, and it's key for autoregulation of blood flow.
Autoregulation, meaning the vessel regulates itself.
Exactly.
Okay.
Think about vital organs like the brain, heart, kidneys. They need a relatively constant blood flow, regardless of whether your overall blood pressure fluctuates quite a bit.
Okay, they need stability.
The myogenic response provides that stability at the level of the small resistance arteries, the arterioles. It's their intrinsic ability to constrict when the pressure inside them increases, and to relax or dilate when the pressure decreases.
So if blood pressure goes up, the vessel pushes back by constricting.
Precisely. The increased pressure stretches the vessel wall. That stretch itself acts as a signal to the smooth muscle cells.
How does stretch trigger contraction?
The stretch is believed to directly depolarize the smooth muscle cell membrane, possibly by opening stretch-activated ion channels. This depolarization opens voltage-gated calcium channels.
Calcium comes in.
Calcium rushes in, intracellular calcium rises, MLCK gets activated, myosin gets phosphorylated, and the muscle contracts. Stretch might also stimulate NSP3 production, adding to the calcium signal.
So the vessel automatically counteracts the pressure increase.
Yes, it helps maintain constant flow despite pressure changes. It also protects the delicate capillaries downstream from potentially damaging high pressures and contributes significantly to the baseline vascular tone that sets overall blood pressure. It's a beautiful local control mechanism.
And again, things can go wrong here. You mentioned cerebral vasospasm after hemorrhage. That sounds like an extreme pathological version of smooth muscle contraction.
It is, unfortunately.
Yeah.
After a subarachnoid hemorrhage, when blood leaks around the brain, breakdown products from that blood, including things like free radicals, act as potent stimuli.
What do they do?
They cause a massive, sustained increase in myoplasmic calcium in the smooth muscle cells of the cerebral arteries. This leads to prolonged MLCK activation and intense, unrelenting contraction vasospasm.
Cutting off blood flow to parts of the brain.
Exactly. Depriving brain tissue of oxygen and nutrients, which can be devastating, leading to stroke or permanent damage.
Can anything be done?
Early on, sometimes vasodilators can help counteract the spasm. But if it persists, a worrying thing happens. The smooth muscle cells themselves can start to change.
Change how?
They can lose their contractile machinery, their actin and myosin, and instead start behaving like fibroblasts, secreting excessive amounts of collagen into the vessel wall.
So the vessel becomes structurally narrowed, not just actively spasming.
Precisely. It becomes a fixed structural constriction that won't respond well to drugs trying to relax the muscle anymore. This highlights the danger of prolonged, inappropriate smooth muscle activation. And interestingly, ROK activity is often elevated in vasospasm, so ROK inhibitors are being explored as a potential therapy here too.
It really underscores how smooth muscle isn't static. It's adaptable, sometimes beneficially, sometimes not. How does it normally grow or adapt to changing demands?
Smooth muscle retains a remarkable capacity for growth and adaptation throughout life, much more so than skeletal or cardiac muscle. During normal development, obviously, the cell numbers increase.
Sure.
But even in adults, if an organ containing smooth muscle faces a sustained increase in workload, the tissue mass increases. We call this compensatory hypertrophy.
Hypertrophy meaning the cells get bigger.
Yes, primarily the existing cells enlarge. A classic example is in chronic hypertension. The arteries constantly face higher pressure, a higher mechanical load. The smooth muscle cells in the artery wall respond by hypertrophying, getting bigger and stronger, making the wall thicker.
Is that purely beneficial?
It's initially compensatory, trying to cope with the load, but it can also contribute to the problem by making the vessels stiffer and narrower. Interestingly, these hypertrophying cells can even become polyploid.
Polyploid, meaning more sets of chromosomes.
Exactly. Multiple sets of chromosomes. This allows them to ramp up protein synthesis dramatically, producing more contractile proteins and other components to increase their size and force capacity. It's a way to boost output without necessarily dividing.
That's quite the adaptation. You also mentioned the uterus as a dramatic example earlier.
Oh, the myometrium, the uterine smooth muscle, is probably the most spectacular example of physiological adaptation. For most of pregnancy, progesterone keeps it relatively quiet, quiescent, with few gap junctions acting more like multi-unit muscle.
Keeping the baby safe inside.
Right. But as birth approaches, the hormonal balance shifts, estrogen becomes dominant, and two amazing things happen. First, the myometrial cells undergo massive hypertrophy, getting much larger and stronger.
Preparing for labor.
And second, a huge number of gap junctions form rapidly between the cells. This electrically couples them all together, transforming the uterus into a powerful, coordinated single-unit muscle ready for the synchronous contractions of labor. It's an incredible transformation.
Absolutely incredible. So that's beneficial adaptation. Where does it cause problems clinically?
Pathological hypertrophy is common. Think about urinary bladder hypertrophy, often seen in older men with an enlarged prostate blocking urine outflow.
The bladder has to work harder.
Much harder against that obstruction. So it's smooth muscle wall hypertrophies to compensate. Initially, this might maintain function, but eventually the thickened, overworked bladder can struggle to contract effectively or empty completely.
Leading to urinary problems.
Exactly. Difficulty urinating, retention. The good news is, if you relieve the obstruction, say by treating the prostate problem, the bladder hypertrophy can often regress and function can improve significantly. It shows the plasticity. But perhaps the most widespread and damaging example of smooth muscle adaptation gone wrong is in atherosclerosis.
Hardening of the arteries. How are smooth muscle cells involved there? They're supposed to be contractile, right?
Normally, yes. But in atherosclerosis, often following injury to the endothelium, the inner lining of the artery, maybe from high blood pressure, smoking, diabetes mellitus, the smooth muscle cells undergo a dramatic change. It's called phenotypic modulation.
Phenotypic modulation, they change their type.
They change their function and structure, yes. They downregulate their contractile machinery, less actin and myosin. Instead, they develop extensive protein-making machinery, like the rough endoplasmic reticulum and Golgi apparatus.
Why would they do that?
They essentially switch from being contractile cells to being synthetic and secretory cells. They migrate from their normal position in the vessel wall into the subendothelial space. They start proliferating, dividing rapidly, and they begin churning out extracellular matrix components.
Like collagen.
Exactly.
Huh.
Collagen, elastin, proteoglycans, the stuff that forms the bulk of the atherosclerotic plaque, the fatty lesion, the fibrous cap. They become key contributors to building the plaque that narrows the artery. It's a fundamental shift in their identity, driven by inflammatory signals.
So they switch from muscle cells to plaque-building cells. That's bad news.
Very bad news. And again, intriguingly, targeting that Rho kinase ROK pathway we discussed. Some studies suggest ROK inhibitors might help reduce atherosclerosis, possibly by affecting endothelial function, inflammation, or maybe even this smooth muscle phenotypic switching. Still research ongoing, but it connects back.
Amazing how interconnected it all is. Okay, we've hit structure, control, adaptation. Let's wrap up with the raw performance, the biophysics. How does smooth muscle measure up in terms of length, tension, and force velocity?
Right, the fundamental mechanics. If you look at the length-tension relationship, it's actually remarkably similar to skeletal muscle when you normalize it. There's an optimal length, L0, where it generates maximum active force, and force falls off if it's shorter or longer than that. A classic bell-shaped curve.
Similar shape. Any key differences?
One difference is that smooth muscle cells can often shorten proportionally more than skeletal muscle cells can from their optimal length. But the really big difference in force generation isn't about having more cross bridges per se.
What is it then?
It goes back to those slow cycling kinetics. Because the myosin heads stay attached to actin for longer periods in smooth muscle, especially in the latch state, a higher proportion of the cross bridges are in the attached force-generating state at any given moment compared to rapidly cycling skeletal muscle.
So even if they cycle slowly, more are pulling at once, leading to high force maintenance.
Exactly. It contributes significantly to their ability to generate high force, especially sustained force, very efficiently.
Efficiency again. Okay, and force velocity. How fast can it shorten against a load?
The general shape of the force velocity curve is also similar to striated muscle. It's hyperbolic, meaning the velocity of shortening decreases as the load or force it's working against increases. Maximum velocity occurs at zero load.
But the speeds are different.
Oh, vastly different. Smooth muscle contraction velocities are much, much slower than in skeletal muscle.
Why so slow?
Primarily because the specific type or isoform of myosin-heavy chain found in smooth muscle has very low intrinsic ATPase activity. It just hydrolyzes ATP, the fuel for contraction, much more slowly. That inherently limits the maximum speed of shortening.
Makes sense.
Doesn't
Slower
really.
fuel burning, slower speed.
But here's where smooth muscle shows its regulatory sophistication again.
Uh-huh.
It can actually alter its force-velocity relationship.
How so? Skeletal muscles is pretty fixed, right? Determined by load and fiber type.
Largely, yes. But in smooth muscle, both the force generated, reflecting the number of active cross bridges, and the velocity of shorting, reflecting the average cycling rate, can vary depending on the level of activation.
Activation level, like how much hormone or nerve signal it's getting.
Exactly. Remember, myosin light chain phosphorylation is key. The level of phosphorylation directly influences both how many cross bridges become active, determining force, and their average cycling rate, determining velocity. Especially with the latch state potentially slowing detachment at lower phosphorylation levels, the muscle can operate at different points on the force-velocity curve depending on the stimulus. It's incredibly tunable.
So it can adjust its speed and force output dynamically based on the signals it receives.
Precisely. It adds another layer of control beyond just on-off.
So bringing this all together after this really deep dive, what does this mean for us, the listener? We've gone from microscopic structure to complex control, efficiency, disease.
Okay, we've covered a lot of ground.
Smooth muscle is clearly this master of adaptation, doing everything from peristalsis to maintaining blood pressure with incredible finesse.
Absolutely. I think the key takeaway is that smooth muscle is far from simple. Its unique features, thick filament regulation, pharmacomechanical coupling, calcium sensitization via pathways like ROAC, the incredible efficiency of the latch state, they all highlight its critical, sophisticated role.
It's not just a background player.
Not at all. It's responding to this constant symphony of signals, electrical, chemical, mechanical, in a highly integrated way. And truly grasping these mechanisms, mastering this information, is fundamental to understanding so much of physiology and pathophysiology.
It really changes how you think about what's happening inside you constantly.
It should.
And maybe the most provocative thought to leave you with today is this. If these incredibly intricate, efficient, and adaptable systems like smooth muscle are operating silently within us, what other profound yet maybe overlooked biological mechanisms are shaping our health every second?
That's a great question.
And how might continuing to take these deep dives, really mastering these foundational processes, unlock even more revolutionary medical insights and breakthroughs down the line?
The potential is definitely there. Understanding the fundamentals is where innovation starts.